Hey everybody, welcome to Traits in Reproduction, Lesson 2.3, Investigating Gene Copies. First things first, we are jumping into the warm-up. The warm-up is going to ask you a question actually about the video that you were supposed to have watched at the end of the last lesson on mutations. It says, in a moment, you will rewatch the video. We're going to watch it again, mutations and new traits. Before watching the video, list one or two questions you may have about mutations. Alternatively, you can list a few things you already know about mutations. So go ahead, do that, and then come on back to me. So, time to rewatch the video. I've attached it again to this lesson. It's also still on my YouTube channel, so you can go check that out. But rewatch the video, and then we're going to answer a couple questions. The questions aren't, I don't know, that focused. What is your reaction to the video? And do you have any questions about the video? I will be asking you about this in class when we come back together, because I'd love to hear your comments on the little cat video. I'm sure you loved it just as much as I did. Next, we are going to be jumping into the sim, investigating gene copies. So let's go ahead and take a look at that. So for investigating gene copies in the sim, yes, you are going to jump in the sim. And we're looking at this question right here. Why do some organisms make one type of protein for a feature and other organisms make two? Well, what do we mean by that? Let's go to this bristle protein example, number one. Anne, she only makes one type of protein. When we look in Anne's cells, can I zoom in? No, I can't zoom in. When we look in Anne's cells, we only see one type of protein for the bristle feature. However, when we look at Leo, we see that Leo is actually making two different types of proteins for the bristle feature. So the question again, why do some organisms make only one type and some make two types? Shouldn't they all just make one type? That's the question. And what you're going to do is you're going to be jumping in the sim, and you're going to find two spiders. Now, you're supposed to do this with partner. You are partner A and you're also partner B. So you're going to do both of these tasks, and I think you will be able to. Choose any feature except for body size. And what you're going to be doing is you're going to be finding two spiders for this feature. One spider that makes only one protein for this feature. One spider that makes two proteins for this feature. Two types of proteins. And then you're going to answer some questions about this. Let's take a look at some of those questions right here. You're going to go ahead and say, hey, okay, here's the feature we observed. The name of the spider with only one protein, the name of the spider that made two different proteins. And why do you think that the spider with only one type of protein made only one type of protein, and the other spider made two types of proteins? You'll write that down right here after you observe those two spiders. Go ahead and do that and come back to me. The next thing that you're going to try to do is you're actually going to try to change your spider's genes so that they both make the same proteins. You can choose, do you want it to look like spider A or spider B? That's totally up to you, but you want to change the genes so that they're both making the same proteins. Now remember, we've done this before. We've changed spiders' genes before, and I think that was in lesson 2.1, and I showed you how to do that at the end of the video because we did that for our homework. So again, go in, find two spiders, and try to change their genes so that they're both making the same proteins, and then come on back to me. Don't forget, in activity number 2 in this lesson, to write down what you did and what you observed. Be specific. I changed the protein or the gene from this to this, and I noticed this. Obviously, I'm not being specific, I'm being general because I don't know what you did, but please do be specific in your explanation. How does changing the spider's genes affect their proteins and resulting traits? Ooh, did you notice that? It doesn't just change the proteins, it changes the traits. Because remember, we already knew this, that proteins are directly related to the traits. The traits that a spider has is directly related to the proteins that it has. And genes affect the proteins. Hey, let's look at this key concept right here. An organism has two copies of a gene for each feature. Did you notice that? Did you notice that when you were changing the spider's genes, there was actually 1 and 2? Okay, well that makes sense because maybe both of those genes are the same. And that would mean that they produce two of the same protein. Maybe they're different and they're producing different proteins. Now, it's for the same feature, but it's going to end up as a different trait. Sometimes. Let's move on. In this diagram, we can see the gene copies in the nucleus of a venom gland cell. The two copies of a gene can be the same or different version. Here's the two copies for venom, and as we can see, these two are the same, but they could have been different, couldn't they? That's what we're going to look at next. The next thing we're going to be doing is we're going to be rereading just part of that hemophilia article. Now, we've already talked about the hemophilia article, what hemophilia is, how it happens, but we are going to reread part of it to help us understand why some organisms make one type of protein for a feature while other organisms make two. You will first reread the paragraph of causes of hemophilia. As you read, pay particular attention to how this diagram and the text fit together. What you're going to be doing is actually in Amplify. In part 1, I want you to, again, read about this diagram, try your best to understand it, and then annotate it. How do I annotate it again? Oh yeah, you hit this plus button right here to add your annotations. Annotate the diagram, please, and then, okay, yeah, that's that part right there. And then when we come back together as a class, we will discuss this. Why do some organisms make one type of protein for a feature and other organisms make two? Some of you already have a really good idea of why this is. But again, we will discuss this. We're going to keep working with this just so we make sure we really understand it. Ooh, look, remember the terms homozygous and heterozygous? I asked you to define these already in the form that I attached when you read this article the first time. To describe what you observed in the sim. Ooh, how can we use these terms to describe what we observed in the sim? Spider A perhaps was homozygous, spider B was heterozygous. Let's take a look at what those definitions are. Homozygous, having gene versions that are the same. Heterozygous, having gene versions that are different. Let's look at an example to help us understand this better. So, the picture up top right here, F1 and F1. These are the genes for, what is this? It doesn't say what these are for. This might be cell flexibility, or yeah. We see the spider has an F1 and an F1. Are those the same or are those different? If they're the same, we would say that this spider is homozygous. Homo meaning the same, zygous meaning something. Let me go look that up. The next picture, F1, F2, same or different? Well, I went to preschool. I know that those are different. And so we would say that that is heterozygous, okay? So we want to start using these terms, homozygous and heterozygous. We want to be familiar with these terms. We want these terms to just naturally come to us when we see gene versions. Oh, these are the same. That's homozygous. These are different. That's heterozygous, okay? So in the reading, we learned that scientists have specific terms to describe these combinations of gene versions. They can either be homozygous, the same, or heterozygous. Thanks for color coordinating. That helps me. Now we want to respond to our investigation question using evidence from the sim and the article using vocabulary words provided. This is also in activity 3, part 3 of activity 3. And there's a nice place for you to go ahead and write your response. Please do try to use all of these terms. Why do some organisms make one protein for each other? You're going to have to talk about genes, gene versions, use those new terms that we have, homozygous and heterozygous, and then come back to me. Last thing is the homework. I really like this homework assignment. I think it's kind of cool because it's asking you to take what you've learned in this unit and try to apply it to something that's seemingly completely different. So in this activity, you will connect what you are learning in the traits and reproduction unit to other science topics. Let's actually go ahead and take a look in Amplify at this homework assignment. Let me read it for you. Think of another science topic you've studied in the past. How does that topic connect to what we are learning about in this unit? be creative. Please be creative. See if you can make connections between genes, proteins, and traits and what you already know about one of the topics below. Hey, you can use one of these topics or you can choose something else. How can you relate this to microbiomes? How can you relate this to metabolism? We've learned about those this year. Or what about any of these things? So tell me what the topic is that you're thinking of, and then, hey, tell me, how does this relate? This is a chance for you to actually be creative, but also in that creativity, you're showing me what you know about this topic because you are describing it in another way. Okay? So have fun with that. And once you're done, we'll see you next time.